Hi guys, Miss Francis here to talk to you about peroxisomes and cytoskeleton. Peroxisomes generate and degrade hydrogen peroxide. We actually did a lab where we used one of the enzymes that are found in peroxisomes. So peroxisomes contain enzymes that transfer hydrogen from various substances into oxygen. When the peroxisomes are doing their various metabolic functions, one of the intermediate products of doing those processes is hydrogen peroxide, H2O2, which is a poison. But the peroxisome has catalase, another enzyme that's going to convert that hydrogen peroxide to water and oxygen, which are both harmless or relatively harmless to humans. Some peroxisomes also break down fatty acids into smaller molecules, which are then transported to mitochondria for fuel, and others detoxify alcohol. Where the structure of a peroxisome includes just a single membrane, I would like for you guys to try and put these in correct size order. So we've got the length of the chloroplast, the thickness of the cell membrane, the length of the mitochondria, E. coli, a eukaryotic cell, and ribosomes. So first would be the cell membrane, the thickness of it. Second would be an organelle known as ribosomes. Third would be our E. coli. Fourth would be the mitochondria. Remember, one of the pieces of evidence for the endosymbiotic theory is that the mitochondria and the bacteria are similar in size and shape. Mitochondria is just slightly larger. Then you've got the chloroplast, and lastly, the largest in this grouping would be our eukaryotic cell. And those are the answers. So now what I'd like to do is discuss the cytoskeleton with you guys. The cytoskeleton is composed of proteins, and its main functions include structure, movement, and locomotion. There are three types of cytoskeleton that we're going to discuss: microtubules, microfilaments, and then down here, intermediate filaments. So let's focus in on the microtubules to begin with. Microtubules are the largest type of cytoskeleton, and they kind of look like a straw. They have a hollow inside, and they are rods. So one of the function is to interact with motor proteins to guide organelles. They kind of act like monorails, where here is the microtubule acting like the monorail for the motor protein, which is then going to move along that microtubule. Another type of microtubule would be centrioles. Centrioles kind of look like churros. If I were to place one churro perpendicular to another churro, that's kind of what the structure of a microtubule known as the centriole looks like. So the centriole will be found in the centrosome, and while both plant and animal cells have centrosomes, only animal cells will have the centrioles, and centrioles are going to play a central role in cell division. They're going to pull the spindle fibers and anchor them as the cell is dividing. Lastly, we have flagella and cilia, which are microtubules, or which are supported by microtubules. And again, flagella and cilia are used for locomotion, for movement. So now let's shift over and let's talk about microfilaments. Microfilaments are the smallest type of cytoskeleton, and you can think of it like a thread-like structure where they are solid rods that are made up of globular proteins. Their main function is to resist tension. For example, actin. While myosin is not a microfilament necessarily, it's a motor protein that moves along the actin. Actin and myosin are often partnered together, and they are responsible for muscle movement. And that would occur in animal cells. In plant cells, actin and myosin are going to work together to do cytoplasmic streaming, which means that within the cytoplasm, there's a circular flow, which speeds up the distribution of materials within the cell. And lastly, you've got intermediate filaments, which are going to reinforce the cell shape and fix the organelle's location. So that's our discussion of peroxisomes and cytoskeleton.